Good evening. Good evening, all of you. Am I audible? Good evening, all of you. Yeah, thank you. Good evening, Korvi Vivek. Good evening, Ravi Shankar, Prakash Mani, Paneerselvam. Yeah, if you're joining the session for the first time, this is Dr. Shanmugapriya. I'm professor and head of the Department of Biochemistry at Government Thoothukudi Medical College. And I'm your Unacademy educator for Biochemistry. And as you all know, today's session is on electrophoresis. Yeah, today we'll be discussing about electrophoresis, chromatography and ELISA. And all these will be discussed with MCQs, with the help of previous year MCQs. Okay. So before we start the session, let me tell you a few facts about Unacademy plans. Plus subscription gives you an access to both live and recorded classes, and it also gives you an access to a question bank with 25,000 plus high yield clinical case based MCQs. Iconic subscription gives you an access to both Unacademy and Preplada platform. Special class features include interactive live sessions. You will be able to participate in polls, and using the raise a hand option, you will be able to interact with the educator one-on-one, and you can get your doubts clarified real time. And these are our toppers. Good evening, Sujit. Good evening, Siddhant. Good evening, Anisha, Armugam. And these are our toppers in the latest NEET PG September 2021. The highest was 694, closely followed by 688. And this is about the test ritual that is happening on a regular basis throughout the week. So first tabular column is about the free test ritual. The second one is about plus batch test ritual. So I strongly recommend that you take up as many tests as possible. And as you all know, following the 4-day session that we are planned for, which will be the study with me session, on Friday you will be having a test, study with me YouTube test series, which will be completely based on these 4 sessions that will be conducted on YouTube. Okay, so try to follow all these sessions and try to revise and attend the test. Okay, and if you have any doubts while you attend the test, you can ask me on my Telegram group. And we have structured batches for all upcoming exams. We have Focus FMG 2022 comprehensive batch, and the next one is Target NEET PG 2022 MCQ discussion batch. And about MCQ discussion batch, one second. Yeah. So, and about the MCQ discussion batch, it has got 2 modules. The first one would be an MCQ discussion format, and the second one will be, good evening Shrish Patak, and the second module will be an ultra fast revision module, which will include previous year question discussion. And this is about the NEET PG subscription plan. The minimum that you can opt for is a 3 months subscription, which would cost you 11,250. But if you use any of the educator's code, you will get 10% off, and you can use my educator code, which is Biochem Life. That will give you 10% off. So that's all about Unacademy plans. Let's start discussing a few MCQs which I planned for you all. So this is the first MCQ. Try to answer this. Electrophoresis separates proteins based on all except charge, molecular weight, size and shape. Can any one of you try answering this? Charge, molecular weight, size and shape. Yeah, the answer is Rahul Jai says it is shape. Janet says it is size. So many of you have answered it as D. Interestingly, it is affected by shape. Okay, so answer is size. Yeah, what is answer? If this is the question that is asked, then the answer would be size. And to understand why it is size, have a look at this. What is the principle of electrophoresis? Electrophoresis is based on the fact that any charged particle will move in an electrical field. Reason is, electrical field is created by a negatively charged electrode and a positively charged electrode. In that case, if the particle is positively charged, where will it move? If it is positively charged, it will move towards negatively charged electrode. If it is negatively charged, it will move towards positively charged electrode. So any charged particle will move in an electrical field, but the rate of migration of a charged particle in an electrical field, the rate of migration or velocity of a charged particle in an electrical field is given by V is equal to Z by F. It's not a complex formula. Try to understand this. Z here refers to the charge. Yeah, Z here refers to the charge, and F refers to the frictional coefficient. And this frictional coefficient is determined by molecular weight and shape. It is determined by molecular weight and shape. Don't you think it's a formula which can be easily understood? True, right? Because what will happen when a particle is more charged? If a particle is more charged, it's going to rapidly move towards oppositely charged electrode. So charge and velocity, how are they related, all of you? Charge and velocity will be directly related. That is why you place charge in the numerator. Okay, now what will happen when the particle is heavy? When the particle is heavy, can it move? No. So weight and velocity will be inversely proportional, and that is why weight is placed in the denominator. That's why I told you F is determined by molecular weight and shape. It's a very simple formula, right? So rate of migration or velocity of a charged particle in an electrical field is given by Z by F, where Z is the charge, F is the frictional coefficient, which is determined by both shape and molecular weight. Okay. So now are you clear about the answer? All of you tell me what is the answer. It is definitely affected by charge. It's definitely affected by molecular weight and shape. So out of these choices, I would go for size. But sometimes the migration is also affected by size. But of these choices, I would go for size. Is that clear? Okay, so the next question is, in serum protein electrophoresis, very good ABCD, very good Shivani, Samaya, good. So the next question is, in the serum protein electrophoresis pattern provided, all of the following proteins are found in the band denoted with a red arrow except. So which is the band that is denoted by a red arrow? They are asking you what are the proteins that can be found in this band. And for this you will have to know first what are these bands, yeah, and then you will have to know what are the proteins that can be found in this band. So can you try answering? DRL says it is D, very good. Ravi Shankar also says it's D, excellent. Manoj Rishi says it's A. Yes, Shrish, you're all right. Answer is antitrypsin. Because antitrypsin, what is the prefix that is given for antitrypsin, all of you? It is alpha-1 antitrypsin. So alpha-1 antitrypsin will be found in alpha-1 band, whereas the band that is denoted here is alpha-2. So I will tell you how to find out what these bands are, okay? But for that you will have to know what is the simple mechanism by which protein electrophoresis is done in the laboratory. Okay, whenever protein electrophoresis is to be done in the laboratory, we use a glass slide. Yeah, we use a glass slide, and on this glass slide we apply a support medium. The support medium will be most commonly agarose. And then towards one end of the glass slide, yeah, towards one end of the glass slide, you apply the serum or plasma, protein of which has to be separated using a micropipette. So the red line which I have shown here represents the line along which you are going to apply the serum or plasma, protein of which has to be separated by electrophoresis. And then we place this glass slide in an electrophoretic tank. Yeah, we place it in an electrophoretic tank and we fill it with an alkaline buffer of pH 8.6. So if you are asking why alkaline buffer of pH 8.6, because only then all the proteins that are present here will sense the alkaline pH. They try to neutralize the alkaline pH. To neutralize the alkaline pH, they give off all their H+. When they give off all their H+, what charge will they carry, all of you? When they give off all their H+, what charge will they carry? They will all now be negatively charged. So the reason behind why you are adding an alkaline buffer of pH 8.6 is to give them all uniformly negative charges. And then you connect this electrophoretic tank to an electrical supply. So close to the point of application is connected to a negatively charged electrode. The opposite side is connected to positively charged electrode. So when you switch on the electric current, because all these proteins are negatively charged, very good Duri, Ravi Shankar, Shrish, Patak, Armuga, Rajesh, good. Because they are all negatively charged, they start moving towards positively charged electrode, and as you all know, the migration here will be affected by number 1 charge, number 2 molecular weight, number 3 shape. And because of variability in all these factors, all your proteins will get segregated into 5 bands. And the band that is present farthest from the point of application, the band that is present farthest from the point of application is an albumin band. So can you all tell me why albumin is the farthest, all of you? You all know the answer, right? Why albumin is the farthest? Because albumin has got the least molecular weight, so it can move the fastest, and it has got the highest negative charges. So albumin is present farthest from the point of application. And after albumin, it is alpha 1 band, and then it is alpha 2, then it is beta, and finally gamma band, which is present close to the point of application. So tell me, what are the 5 bands present in protein electrophoresis? It's albumin, alpha 1, alpha 2, beta and gamma. So what are the proteins that are present in alpha 1 band? Give me names of those proteins which have a prefix alpha. What is the first protein I told you? It is alpha 1 antitrypsin. Second one is alpha fetoprotein, and then transthyretin. So tell me, what are the 3 proteins that are present in alpha 1 band? This can be asked. So alpha 1 antitrypsin, alpha fetoprotein and transthyretin. Now what are the proteins that are present in alpha 2 band? Very good, Manasa. Very good, Janet. Good, Shrish. So what are the proteins that are present in alpha 2 band? Any protein with a prefix alpha 2? Alpha 2 macroglobulin. Alpha 2 band has got alpha 2 macroglobulin, ceruloplasmin and haptoglobin. Alpha 2 band has got alpha 2 macroglobulin, ceruloplasmin and haptoglobin. And then it is beta band. The next one is beta band. Beta band has got beta 2 microglobulin and transferrin. Beta band has got beta 2 microglobulin and transferrin. Now what is the last band? The one which is closest to the point of application is gamma band. And what are the proteins that you expect in gamma band? Gamma globulins. What are gamma globulins? Immunoglobulins and fibrinogen. Okay, immunoglobulins and fibrinogen. Gamma band has got immunoglobulin and fibrinogen. Is that clear, all of you now? So can we have a look at the band that I showed you, the pattern which I showed you? And whenever you're shown an electrophoretic pattern, step number 1 is find out where is the point of application. Here do you see that they have noted that this is the point of application. Okay. So this is the point of application, and the band that is farthest from the point of application, this will be albumin. And after albumin it is alpha 1. This is alpha 2, this is beta, and the smeared band that you find here, the smeared band that you find here is gamma band. Is it clear? This is how you identify. First find out where is the point of application, and the band that is farthest from the point of application will be an albumin band, and then alpha 1, alpha 2, beta and gamma. Okay. So the question that is asked you is about alpha 2 band. So can you all type what are the proteins that are present in alpha 2 band? I said alpha 2 has got alpha 2 macroglobulin, ceruloplasmin and haptoglobin. So what is the answer? The answer is antitrypsin, because antitrypsin is present in alpha 1 band. Clear? So the next question. Good, Shivani. So the next question is the clotting factor that is present in serum is. Which is the clotting factor that is present in serum? Factor 12, 10, 5, 7. Can any one of you answer? Good, Rahul. Rahul says it's D. Can any one of you? Yes, factor 7. Janet, very good. So the answer is factor 7. Good. Okay. So let's try to understand what is the difference between a serum and a plasma. Tell me, what is a serum? If you collect a First, let me tell you what is plasma. Okay. So if you collect a blood sample, if you collect a blood sample in a tube in which you have placed an anticoagulant, what separates above is called as plasma. Isn't that easy to remember? If you have placed an anticoagulant, what separates above is called as plasma. So what is serum? If you collect a blood sample in a simple tube, yeah, the negative charges of the tube will stimulate in vitro clotting. And after in vitro clotting, you know, a hard rock clot is formed. After a hard rock clot is formed, what separates above is called as serum. So if you know that serum is something which separates after clotting, it means serum does not have clotting factors. Do you all understand this? Serum does not have clotting factors in the sense it does not have fibrinogen. And though we say it does not have clotting factors, it can still have factor 7. The reason is factor 7 is involved in extrinsic pathway. Don't we all know this? Yeah, factor 7 is involved in extrinsic pathway. Whereas negative charges of the glass or the tube will stimulate factor 12 becoming 12A, right? 12 becomes 12A, 11 becomes 11A, 9 becomes 9A. It's basically intrinsic pathway. All of you please remember this. In vitro clotting involves intrinsic pathway, whereas factor 7 is involved in extrinsic pathway, so that is preserved. So tell me which factor is present in serum, all of you? Which clotting factor is present in serum? It is factor 7. Okay. So now the next question is, I have given you enough foundation for you to answer the next question. Okay. So I'll be happy if you can all answer this question. The preferred sample for electrophoresis for diagnosis of multiple myeloma is? The preferred sample for electrophoresis for diagnosis of multiple myeloma is? What is the answer, all of you? Very good. Good Janet, good Anisha. So it is always serum. Yeah, we call it as serum protein electrophoresis. Please remember this. We call it as serum protein electrophoresis. And what did I tell you about the difference between serum and plasma, all of you? What is the difference between serum and plasma? I said serum does not have fibrinogen. And fibrinogen forms a band in which region? Fibrinogen forms a band in the gamma region. And what is that you know about multiple myeloma? Multiple myeloma is a plasma cell neoplasia wherein there is monoclonal proliferation of plasma cells. That is, there is excess production of one clone of antibodies. Yeah, there is excess production of one clone of antibodies. In that case, which band do you think will get affected in multiple myeloma, all of you? Excess production of antibody. Antibodies are found in which band? Gamma region. Yes. So how will the gamma band look? In multiple myeloma, will the gamma band be intense or dull? Just answer this now. Will the gamma band be intense or dull? Because the concentration of antibodies is more here, it is going to be an intense band. And will the band be broad or narrow, all of you? Will the gamma band be broad or narrow? Yes, it is intense. Very good, Janet. It is going to be a narrow band. Yeah, please try to understand this because generally students think when the concentration increases, it will be a broad band. No, it is going to be an intense narrow band that is seen in the gamma region, which is called as M band of multiple myeloma. So why will it be narrow and not broad? It is very simple. What did I tell you? The migration is affected by charge, molecular weight and shape. And when it's a monoclonal proliferation of antibodies, all these antibodies will have the same shape, same molecular weight and same charge. So they'll all move in one line, they'll all stand in one line. Okay, so how will the band be? The band will be narrow and intense in the gamma region, which is called as M band of multiple myeloma. Is that clear, all of you? What if you see a broader band? What if you see a broader band in the gamma region? It means it is polyclonal proliferation. When do you expect a polyclonal proliferation? Only when there is chronic infection. Yeah, for example, if the person is suffering from tuberculosis, if you are running protein electrophoresis of a patient of tuberculosis, there is polyclonal proliferation of antibodies, and in that case you will find a smeared band. The band will look like a smear in the gamma region. Is that clear? So now answer this question. The preferred sample for electrophoresis for diagnosis of multiple myeloma. So you know in multiple myeloma you expect a band in the gamma region Yeah, so you have to run serum protein electrophoresis, because if by mistake if you run plasma protein electrophoresis, plasma has got fibrinogen and that forms a band in the same gamma region where you expect M band of multiple myeloma. So fibrinogen band will obscure the M band of multiple myeloma. Do you all understand this? So never run plasma protein electrophoresis. It has to be serum protein electrophoresis. So what we do in the laboratories, after we receive the blood sample, before we run it through electrophoresis, we just tilt the blood sample and check. Only when you find a hard rock clot, only when we find a hard rock clot in the bottom, we will use a supernatant. Otherwise, we'll ask for a repeat sample. So what is the preferred sample, all of you? The preferred sample is serum. Okay. What is the abnormal pattern that is seen in the electrophoretic pattern provided? So they've given you an electrophoretic pattern, okay, and this is the band that is shown to you. They're asking you what band is this. But what did I tell you? Whenever you're shown an electrophoretic pattern, you should find out where is the point of application. Though they have not marked the point of application here, they have written it as a cathode end. Cathode is negatively charged. And then I tell you, you always apply serum or plasma close to the negatively charged electrode. So the point of application should be somewhere here. Yeah, the point of application should be somewhere here. And from the point of application, exactly opposite, which is the last band from the point of application I told you. The last band will be, some of you have answered it as A. Akila says it's B. Shrish says it's C. Okay, so I will tell you the answer in a minute. Okay, so try to understand this. So this is the point of application, and farthest from the point of application, this is the albumin band. Okay, and then there is an alpha 1 band, alpha 2, beta, and this band is nothing but gamma band. The band that is shown to you is gamma band. And this gamma band is intense. Yeah, it is intense and narrow. So what will you call it as? Narrow intense band in the gamma region is called as M band of multiple myeloma. Okay, instead of you finding a narrow intense band, suppose you see a broad intense band like this. Yeah, broad intense band like this, then you will say it is a broad intense band, which is a feature of what, all of you? Broad intense band is a feature of chronic infections. Okay, and when will you find relative prominence in alpha 2 band? When will you find relative prominence in alpha 2 band? Can you all tell me what are the proteins that are present in alpha 2 band, all of you? Alpha 2 band has got alpha 2 macroglobulin. Yeah, it has got macroglobulin. So tell me what happens in nephrotic syndrome. Yeah, relative prominence of alpha 2 band is a feature of nephrotic syndrome, because nephrotic syndrome is nothing but leaky glomerulus. So glomerulus has started leaking all small proteins. So which protein alone will be retained? The macroglobulin, alpha 2 macroglobulin alone will be retained in the circulation. And because glomerulus has lost all small proteins, if you look at the electrophoretic separation, it will not look bright blue like this. It will be a very dull electrophoretic separation in a patient of nephrotic syndrome, indicating that there is a reduction in the serum total protein concentration. So in a dull background, there will be relative prominence of alpha 2 band alone, because alpha 2 band has got alpha 2 macroglobulin, which alone has been retained in the circulation. So in nephrotic syndrome, what is the band that you see, all of you? In nephrotic syndrome, it is relative prominence of alpha 2 band. Okay, and then beta gamma fusion. When do you find beta gamma fusion? In the case of decompensated liver disease. Because what happens in decompensated liver disease? Liver stops producing albumin. And you know, albumin is the major determinant of oncotic pressure. And when the blood oncotic pressure decreases, to compensate for the reduction in the oncotic pressure, plasma cells will start producing more and more antibodies. Yeah, so polyclonal proliferation of B cells is what you find. And when there is polyclonal proliferation, what will happen to gamma band? Yeah, gamma band will be very wide. and the wide gamma band comes and fuses with the beta band, and that is called as beta gamma fusion of decompensated liver disease. So all of you have a look at this tabular column. This tabular column will tell you about significant electrophoretic patterns that you can observe in serum protein electrophoresis. And serum protein electrophoresis can be used to make a diagnosis of multiple myeloma, whereas it can be used as a supportive evidence for nephrotic syndrome and for decompensated liver disease. And what is that you observe in multiple myeloma? A narrow intense band in the gamma region, which is called as M band of multiple myeloma. And what do you find in nephrotic syndrome, which can be used as an add-on evidence? In nephrotic syndrome, all other proteins are lost, so there is relative prominence of alpha 2 band. In decompensated liver disease, you find beta gamma fusion. Okay. Why alpha 2 macroglobulin does not leak is Rahul Jai's question. It is because macroglobulin size is larger. In nephrotic syndrome, when there is leaky glomerulus, only smaller proteins get lost, larger proteins will be retained. Predominantly albumin gets lost. So larger proteins like alpha 2 macroglobulin will be retained. Clear? So that's about, is that clear, Rahul Jai? So that's about that question. Now about the next question. SDS-PAGE separates proteins based on this. I have seen at least 4 times in NEET and All India. Yeah, SDS-PAGE separates proteins based on. What is your answer? Very good, Shrish. Very good, Janet. Very good, Shrish again. Yes. What is answer? Yes, molecular weight. Yeah, all of you try to memorize this. SDS-PAGE separates proteins based on molecular weight. So before I start telling you what SDS-PAGE does, yeah, let me tell you what happened, or let us recall what we discussed about what are the factors which determine mobility of proteins in an electrophoretic field. What are the proteins I said? The factors which affect electrophoretic mobility will be number 1 charge, I told you, right? Number 1 charge, number 2 is shape, number 3 is molecular weight. So there are 3 factors which affect electrophoretic mobility. But if you have multiple factors determining electrophoretic mobility, that mobility cannot be used to characterize a protein. Yeah, this mobility cannot be used to characterize a protein. For example, if you see a protein to move fastest, yeah, will you immediately say that this protein has got the least molecular weight? That cannot be your inference, because this protein could have moved faster because it has got highest negative charges. So you're not able to comment on its molecular weight. Okay. Similarly, you see that a protein does not move at all. If it does not move at all, will you immediately say it does not have charges? No, because the protein might not have migrated because it's very heavy. So you're not able to comment on its charge as well. So if you have multiple factors determining mobility like this, you won't be able to characterize a protein based on this mobility. And that is why, to rule out multiple factors and to have only one factor determine mobility, we add SDS. And what is SDS? SDS is sodium dodecyl sulfonate. Yeah, SDS is sodium dodecyl sulfonate. And this sodium dodecyl sulfonate is a polyanionic detergent. Yeah, what is it basically? It's a polyanionic detergent, and because it's polyanionic, it removes charge effect. I will tell you how it removes charge effect in a minute. For now, just remember it removes charge effect. And because it's a detergent, detergent is something which removes all higher orders of structure. It removes secondary structure, it removes tertiary structure. It allows a protein to have only primary structure. And you must have learned, and I have also taught you when I told you about structure of proteins in special classes, that it is a secondary structure and tertiary structure which gives a protein a unique shape. And because it's a detergent, because it removes all higher orders of structure and retains only primary structure, it removes shape effect, because all proteins are now spherical. Yeah, if it's only primary structure, then all proteins are spherical. So you've removed shape effect. So what is the only factor that is left out? I told you there are 3 factors which determine mobility, right? What are the 3 factors I told you? Charge, shape and molecular weight. Because it's a polyanionic detergent, it has removed charge effect, it has removed shape effect. So what is the only factor that is left out? It is molecular weight. And that is why we always say that SDS-PAGE separates proteins purely based on molecular weight. Now I'll quickly help you understand how SDS removes the charge effect. Okay, so look at this electrophoretic separation. This is the point of application. Yeah, this is the point of application. So the band that is farthest from the point of application is albumin band. And the one that is present closest to the point of application is gamma band made up of immunoglobulins. So why do you think albumin moves so fast when compared to immunoglobulin? It is because albumin has got the highest negative charges. Human serum albumin carries 6 negative charges when dropped in a pH of 8.6. Whereas immunoglobulins carry only 2 negative charges. Yeah, immunoglobulins carry only, on an average, they carry only 2 negative charges. So what magnitude more negative charges do you find in albumin when compared to immunoglobulin? Answer this. What magnitude more negative charges do you find in albumin when compared to immunoglobulin? 3 times. Albumin has got 3 times more negative charges, and that is why you see the difference in mobility. Now what happens after you treat all these proteins with SDS? Yeah, SDS adds 1000 negative charges to all the proteins. So how many negative charges are there with albumin? It is 1006. How many negative charges are there? Magnitude is not difference. It is number of times, Duri Ravi Shankar. Okay. So how many negative charges are there in immunoglobulin? It is 1002. So 1006 is to 1002. Yeah. 1006 is to 1002 is as good as 1 is to 1. Do you understand this? That is why you don't see any difference caused by charges after you treat the protein with SDS. Have you all understood it? Yes. So tell me SDS-PAGE. If you are asked about SDS-PAGE, what should be your answer? You should remember that it separates proteins based on molecular weight. And if you are asked why does it separate proteins only based on molecular weight, your answer should be because it's a polyanionic detergent. And it removes charge effect, it removes shape effect, so it separates proteins purely based on molecular weight. Okay. So this is the next question. And this has been asked sometime back in INI-CET. Yeah, AIMS question it is. So let's try to read it. I'm not going to read the initial few statements. The initial few statements tell you that they have found out something called as anti-PD-1 antibody. Have you heard of anti-PD-1 antibody? It has got anti-neoplastic features. Okay, so they found out anti-PD-1 antibody. They extracted it from culture. And they have run it through SDS gel electrophoresis. They have run anti-PD-1 antibody through SDS gel electrophoresis. The pattern of the protein migration is provided here. M is the protein ladder separation. Do you see M here? Yeah, this is protein ladder. What is a protein ladder? It's a combination of known protein of known molecular weight. Lane 1 and lane 2 are non-reducing and reducing lanes respectively. So lane 1 they say is non-reducing and lane 2 is reducing lane. Okay, lane 1 is non-reducing, lane 2 is reducing lane. And the question is, what is your comment on this protein? Is it a monomer? Is it a dimer? Is it a trimer? Is it a tetramer? Can anyone try to answer this? Do you think it's a monomer? Do you think it's a dimer or a trimer or a tetramer? Siva Reddy says it's choice A. Anyone else? You don't have to be right always. Just make an attempt. Ravi Shankar, Duri Ravi Shankar thinks it's trimer. Manasa Pulala thinks it's A. Janet thinks it's a trimer. So it's either monomer or trimer is the answer that I see continuously. So let me tell you what is reducing lane and what is non-reducing lane. Okay, non-reducing lane is you treating protein with just SDS. Reducing lane is you are treating the protein with SDS and mercaptoethanol. Do you understand this? Reducing lane is where you treat the protein with not only SDS but also with mercaptoethanol. And what is mercaptoethanol? It is a reducing substance. Yeah, it reduces disulfide bridges. And don't you all know that in all proteins, individual subunits are connected together by disulfide bridges, all of you? Yeah, individual subunits are connected together by disulfide bridges. So when you treat the protein with mercaptoethanol, the disulfide bridge is broken and becomes sulfhydryl group. Okay, and so individual units will become free. Do you understand this? Individual units will become free. Now with this basic introduction, look at the picture. So lane 1 is non-reducing. So this is the whole protein. So the whole protein has moved and stopped here. So what is the molecular weight? This whole protein has stopped here. This tells you that the whole protein's molecular weight is 150 kilodalton. After treating the protein with mercaptoethanol, it is giving rise to 2 bands. Yeah, one corresponding to 50 kilodalton, the other subunit corresponds to a molecular weight 25 kilodalton. So now tell me the answer. Will any one of you say it's a monomer? Will any one of you say it's a monomer? Definitely it's not a monomer, right? Had it been a monomer, even after treating with mercaptoethanol, there should have been only 1 band. So it is not a monomer. Does any one of you think it's a dimer? All of you think it's a dimer except Vivek. So Ragul, Duri Ravi Shankar, Janet, you all think it's a dimer. So it is not a dimer because had it been a dimer, the individual subunits molecular weight when you add, it should be equal to the whole protein. What is the whole protein's molecular weight? I said it is 150. And individual subunits molecular weight is 50 and 25. Do you think 50 plus 25 is 150? No. So what is it? It's actually a tetramer. Yeah, it is not a dimer because the sum of the molecular weight of individual units is not equal to the whole protein's molecular weight. So it's not a dimer. So what is the answer, all of you? It's a tetramer. It's a tetramer made up of 2 subunits. Each subunit's molecular weight is 50 kilodalton. So 50 into 2 will be 100. Another 2 subunits molecular weight 25 kilodaltons. 25 into 2 will be 50. So totally it is 150. Is that clear, all of you? Yeah. So this is reducing type of SDS-PAGE. So this reducing type of SDS-PAGE will help you understand how many subunits are there in a protein. And that is why in the special class which I conducted probably a week back on protein structure, I said quaternary structure of a protein is studied using reducing type of SDS-PAGE. Now do you understand it? Yeah, reducing type of SDS-PAGE, when you use mercaptoethanol, you clearly understand how many subunits are there and what is the molecular weight of every subunit. Okay, so the answer is it's a tetramer. Now the next question, the amino acid which moves the fastest in paper chromatography is, yeah, the question is the amino acid which moves the fastest in paper chromatography is. Can you all try answering? As I always say, you don't have to be right, but please attempt every MCQ that is shown to you. Is it glutamic acid or aspartic acid, or is it histidine or tryptophan? Vivek thinks it's histidine, okay. DRL thinks it's again the same. Akhilesh thinks it's glutamic acid. Good, I see variety of choices here. And the answer is tryptophan. Okay, answer is tryptophan. So let me tell you the basics of paper chromatography because this has been frequently asked. Okay, the exact question I've seen. The amino acid which moves fastest in paper chromatography is. So before I start telling about paper chromatography, let me tell you just 2 words about chromatography. So chromatography is a separation technique wherein you separate solutes. Yeah, wherein you separate solutes using 2 phases. One is a mobile phase, the other one is a stationary phase. So depending upon whether a solute has got an affinity to mobile phase or to stationary phase, a solute will either move faster or the solute will get retarded. Yeah, if a solute has got an affinity to mobile phase, a solute will move. If the solute has got an affinity to stationary phase, it will get retarded. That way, what have you done? You have separated solutes. Do you all understand this? So whenever you're asked about chromatography, just remember it's a separation technique where you separate solutes depending upon whether the solute has got an affinity to a mobile phase or to a stationary phase. Okay. And the simplest chromatographic separation is paper chromatography. And as I always say, it is obsolete. Yeah, paper chromatography is obsolete because we have now progressed to HPLC. And the only place wherein paper chromatography is still in use is, the only place wherein paper chromatography is still in use is academic purposes. MD Biochemistry final exam practical, it carries 50 marks. Otherwise I don't see any significance behind paper chromatography. But because it has been asked, I will be happy if you try to understand it. Okay. So what we do in paper chromatography, we take Wattman paper number 1. We take Wattman paper number 1. We cut this paper into dimensions of 30 cross 35 centimeter. So the base will be 30 centimeter, the height will be 35 centimeter. And 5 centimeter from the base, we mark a line using a pencil. And on this line, we mark a dot. And on this dot, we apply the amino acid mixture which has to be separated using a capillary tube. Do you understand this? On this dot, we apply the amino acid mixture which has to be separated using a capillary tube. And then we take this paper and we tie the paper. If you bring the edges together and you tie it with needle and thread, we do that. Okay. So you take the edges of the paper, you tie it with the needle and thread, and this is how the paper would look. Okay. And then we place this paper on a petri dish. You must have worked with a petri dish, right? So we place it on a petri dish and we fill this petri dish with a solvent that is used for amino acid separation. When they can ask you which amino acid moves the fastest, they can even ask you this. So what is a solvent that is used for amino acid separation? It is a combination of N-butanol, acetic acid and water. So tell me, what is the solvent that is used for amino acid separation? It is N-butanol, acetic acid and water. So the moment I place the paper on the petri dish, because this petri dish has got water, because the petri dish has got water, and because paper is polar and water is also polar, water will form a thin layer on the paper. Do you understand this? Quickly, water will form a thin layer on this paper, and then N-butanol and acetic acid will start moving on the paper. N-butanol and acetic acid both are non-polar, still they start moving on the paper because of capillary action. So when these non-polar solvents move along the paper, when they cross this dot, when you place amino acid mixture, they will try to take along with them few amino acids. Which amino acid do you think they will take along? Will they take along non-polar amino acid or will they take along polar amino acid? They are non-polar, right? So hydrophobic interaction. So they will be able to interact only with non-polar amino acid. So as N-butanol and acetic acid start moving on the paper, along with that non-polar amino acids will also move. So they get separated into multiple dots depending upon the polarity. The most non-polar, moves the fastest and it stays away from the point of application. The most polar amino acid will not be able to move at all, so it will be present close to the point of application. That way you will be able to separate amino acids based on polarity. So in paper chromatography, there are just two carry home messages. Listen to this carefully. In paper chromatography, there are just two carry home messages. The first one is you should know which is the stationary phase and which is the mobile phase. So tell me which is the mobile phase. What moves? I said N-butanol and acetic acid move. And I said both N-butanol and acetic acid are non-polar. So it is basically a non-polar mobile phase. So only non-polar amino acids move. And which is the stationary phase? The stationary phase is paper. The stationary phase is paper and water. So stationary phase is polar. So polar amino acids will be retarded. Is that clear? So if they ask you in paper chromatography which amino acid moves the fastest, it means what are they asking you? Which will move the fastest? They are asking you of the following which is the most non-polar. Okay. Now look at these choices and tell me the answer. It is definitely not glutamic acid because glutamic acid is glutamate. Yeah, any acid will be negatively charged. Anything which is charged is polar. Aspartic acid is also polar because it's negatively charged. And I've told you, when I told you about classification of amino acids, I said histidine is a basic amino acid. It is polar, positively charged, so it is also polar. So what is the mnemonic I gave you for basic amino acids, all of you? What is the mnemonic I gave you for basic amino acids? HLA. What does HLA stand for? H stands for histidine, L stands for lysine, A stands for arginine. Okay, so histidine, lysine and arginine are the basic or positively charged amino acids. So tell me, what is the answer? The answer is tryptophan, which is very non-polar because it's an aromatic amino acid. Yeah, tryptophan is an aromatic amino acid. Clear? So that moves the fastest. So why have I included this question? Did you see in the NEET PG 2021, they asked you the most widely used technique for estimating HbA1c. Yeah, the most widely used technique for estimating HbA1c. What is answer, all of you? Glycated hemoglobin is widely estimated by ion exchange chromatography. To be very specific, it is cation exchange chromatography. Yeah, HbA1c is estimated widely by what? Cation exchange chromatography. So when they can ask you this, they might ask you in detail about ion exchange chromatography. Okay. So which of the following is true about ion exchange chromatography? What is your answer, all of you? What is answer? Vivek says it's C. Good. Any other answer? Try your luck, please. Do you think it's A or B or C or D? Yeah, as Vivek has answered it, is choice C. DEAE column is an example of anion exchange chromatography. So let's try to understand about ion exchange chromatography. The first fact is ion exchange chromatography is of two types. One is cation exchange, the other one is anion exchange. Yeah, listen to this carefully. You use cation exchange chromatography when the protein of interest to you is a cation. Do you understand this? You use cation exchange chromatography when the protein of interest to you is a cation. Example is HbA1c. HbA1c is more positively charged when compared to non-glycated hemoglobin. Okay, so how do you estimate HbA1c, all of you? It is estimated by cation exchange chromatography. And when the protein of interest to you is a cation, always in chromatographic separation, yeah, always in chromatographic separation, anything which is of interest to us will be held on to the stationary phase. Okay, so that anything which we don't want, which we consider as impurities, will be allowed to first descend down. And after you have removed all impurities, you will get back what you want from the stationary phase by a technique called as elution. Have you all understood this? So in there are many types of chromatography. For example, ion exchange chromatography, we have affinity chromatography, we have so many types. But irrespective of the type, in all chromatographic techniques, anything which is of interest to us will be first held on to the stationary phase. So then anything which you do not want, which you consider as impurities, will descend down first. So in cation exchange chromatography, I said you use cation exchange chromatography only when the protein of interest to you is a cation, which means cation should be held on to the stationary phase. Yes or no? Cation should be held on to the stationary phase, for which the stationary phase should be an anion. Yeah, for which the stationary phase should be an anion. So in this case, you use as a stationary phase a column. In cation exchange chromatography, use as a stationary phase a column which is filled with synthetic polymer beads which are covalently attached to negatively charged groups. Yeah, they will be negatively charged groups. Example, any acidic group can be added. Give me examples for acidic group: carboxyl group, sulfate group, phosphate group. So any acidic group, any negatively charged group can be added. So example for cation exchange column is carboxymethylsaccharose. So have a look at this. So this is the basis of cation exchange chromatography, wherein the protein of interest to you is a cation, and to hold the cation onto the stationary phase, the stationary phase is made up of beads which are covalently attached to negatively charged groups like carboxyl groups. So C for C, cation exchange is carboxymethylsaccharose. Okay. So have a look at this. So I told you, right, in cation exchange, this is a column. Yeah, this is a column. You must have all worked with the biuret, right? Pipette biuret. Yeah. So this the biuret is used here as a column, and this column is filled with synthetic polymer beads which are covalently attached to negatively charged groups. So when you pour the complex protein mixture, anything which is positively charged alone will be held back, and anything which is negatively charged will be repelled and it descends down. Okay, so after you removed all that you don't want, how will you get back what you want from the stationary phase? Only when you get back what you want from the stationary phase, you can quantify it. So how will you get what you want from the stationary phase? By a technique called as elution. Yeah, by a technique called as elution. And in cation exchange chromatography, for elution, we pour an alkaline buffer. Yeah, in cation exchange chromatography, for elution, you pour an alkaline buffer, because the moment you fill this column with an alkaline buffer, all these proteins will try to resist the alkaline pH. To resist the alkaline pH, they will give off their H+. And once they give off their H+, they are no more positively charged. They cannot be retarded onto the stationary phase, so they come down along with the buffer. Yeah, so this is about cation exchange chromatography. Now I want you to answer about anion exchange. Can you all type it in the chat box? The live chat should be active, please. So when will you use anion exchange chromatography, all of you? You use anion exchange chromatography when the protein of interest to you is an anion. And what should you do to the protein of interest? Always the protein of interest should be held onto the stationary phase. So you have to hold anion onto the stationary phase. How can you hold? Very good. How can you hold anion onto the stationary phase? Only when the stationary phase is a cation. It has to be positively charged. Example, quaternary ammonium groups. Yeah, example is quaternary ammonium groups, N+. An example for that is diethyl aminoethylsaccharose. Example is diethyl aminoethylsaccharose. Here amino means it is N+. So what is the example for cation exchange? C for C, carboxymethylsaccharose. Example for anion exchange is diethyl aminoethylsaccharose. And in cation exchange, I said for elution you use an alkaline buffer. So in anion exchange you will be using an acidic buffer. Yeah, you will be using an acidic buffer. So can you read all the choices, or I will read all the choices? Tell me if these choices are true or false. Look at choice A, cation exchange chromatography uses cations in the stationary phase. No. To retard cations, the stationary phase should be an anion. So this is false. Okay. In anion exchange chromatography, alkaline buffer is used. This is also false. Right. So only in cation exchange you use an alkaline buffer. Diethyl aminoethyl column is the example for anion exchange. I told you, right? C for C. Cation exchange example is carboxymethylsaccharose. Anion exchange example is diethyl aminoethyl column. So this is true. Yeah, so which is the true statement? True statement is choice C. Good, Rahul. In cation exchange chromatography, acidic buffer is used. No, alkaline buffer. Okay, this now I think you will all be able to answer this without a doubt. The most widely used technique for estimating HbA1c is, what is answer? The most widely used technique for estimating HbA1c without a doubt is cation exchange chromatography. Very good. But I want you to know that HbA1c can be estimated by all these techniques. You can estimate HbA1c by cation exchange chromatography, by affinity chromatography. It is called as boronate affinity. Yeah, it is called as boronate affinity chromatography. It can be estimated by electrophoresis. It can also be estimated by immunoturbidimetry. Yeah, it can also be estimated by immunoturbidimetry. So what are the 4 methods by which you can estimate HbA1c? Cation exchange chromatography, boronate affinity chromatography, electrophoresis and immunoturbidimetry. Of the 4, which is the most widely used? The most widely used will be cation exchange. Okay. So this is the last question for tonight. Identify techniques A and B. Can you all try answering this? Identify techniques A and B. Sandwich immunoassay, competitive immunoassay, competitive and sandwich, direct, indirect, indirect, direct. Yes, what is answer? Has Vivek answered it, or is it the answer for the previous question? I want all of you to try answering. Quick. Okay. I will tell you. Duri Ravi Shankar thinks it's B. Rahul Jay thinks it's A. Okay. Answer is choice A, all of you. Good, Vivek. Answer is choice A. It is sandwich and competitive immunoassay. But to be able to understand it, you will have to know few terms. Yeah, few terms in immunoassay. The first term is what is a capture antibody. The second one is what is a signal antibody. So what do you think is a capture antibody? It's an antibody which holds the analyte. Yeah, it's an antibody which holds the analyte to the measurement surface. It captures the antigen to the measurement surface. That's why it's called as capture antibody. What is a signal antibody? This is an antibody which is attached to the label. Do you understand this? What's a signal antibody? This is the antibody which is attached to label. Now there are 2 other terms that you will have to know. One is primary antibody, the other one is secondary antibody. What is a primary antibody? It is an antibody which is directly attached to antigen. Primary antibody is an antibody that is directly attached to antigen. What is a secondary antibody? It is an antibody that is directed against an antibody which is attached to antigen. Do you understand this? What's a secondary antibody? It's an antibody against another antibody. Have you understood all these terms? So capture antibody captures the antigen to the surface. Signal antibody will have the label that is attached. And in ELISA, what is ELISA? It is enzyme linked immunosorbent assay. In ELISA, which is the label? The label is the enzyme. Now, what are primary antibodies and what are secondary antibodies? Primary antibody is directly attached to antigen. Secondary antibody is attached to another antibody. Okay. Now, look at this image. Yeah, this is the first image that was shown here. And in this image, the green color structure. Yeah, the green color structure is the analyte that we are trying to measure. That is the antigen. And this antigen is captured by what? This is the measurement surface. So this antigen is captured by captured antibody. Yeah, the blue color structure is capture antibody. And do you see labels here? Yeah, so this antibody is attached to label. So these antibodies are called as signal antibodies. And what you find here is, don't you find that here the antigen is sandwiched? Yeah, don't you see here that the antigen is sandwiched between capture antibody and signal antibody? Yes or no? I am so smart. Thank you, Vivek. I am happy. Yeah, so don't you see that the antigen is captured, the antigen is sandwiched between capture antibody and signal antibody. In that case, what will you call it as? You will call it as a sandwich immunoassay. Okay. Whereas the next image that is shown to you is this. So even in this image, which is the antigen or the analyte? The same green color structure is the antigen or the analyte. And which is the capture antibody? The blue color structure which captures it to the surface area is called as the capture antibody. Now what is a signal antibody? You don't have an antibody here. You see here that there is something which has got the label, and the label is attached to another antigen. So it is a signal antigen, and this signal antigen competes with the analyte. Do you see that here? The signal antigen is competing with the analyte to go and bind to the capture antibody. So there is a competition between unlabeled antigen. Yeah, there is a competition between unlabeled antigen or the analyte and the labeled antigen or the conjugate. Yeah, there is a competition between unlabeled antigen or analyte and the labeled antigen or conjugate. And that is why it is called as competitive immunoassay. And in competitive immunoassay, if you see more signal, what does that mean? In competitive immunoassay, if you see more signal, it means the labeled antigen has succeeded. It means the labeled antigen has overcome the binding of the analyte, which means analyte concentration is low. So if you see more signal in competitive immunoassay, it means analyte concentration is low. And that is why this graph is shown here. Yeah, the image that was shown in the question has got this graph, wherein as the concentration of the analyte increases, you see that the signal decreases. So if you see this kind of a graph, then you can close your eyes and say it is a competitive immunoassay. Whereas what happens in a sandwich immunoassay? In a sandwich immunoassay, as and when the antigen concentration increases, more and more signal will be seen. As and when the antigen concentration increases, more and more signal will be seen. So this is how you will see a graph. As the concentration increases, the signal increases. So if you see a directly proportional graph, it is sandwich immunoassay. If you see an inversely proportional graph, then that is competitive immunoassay. So look at image A, all of you. Image A, the antigen or the analyte is sandwiched between capture antibody and signal antibody. So this is sandwich immunoassay. And here as the concentration increases, the signal increases. Here there is a competition between unlabeled antigen and labeled antigen. So B is a competitive immunoassay. And you see that as the concentration increases, the signal decreases. So A is sandwich immunoassay and B is competitive immunoassay. Is that clear, all of you? Yeah. So do you have any doubts so far in electrophoresis chromatography and in ELISA? If you have any doubts, you can type it in the chat box or you can ask me on the Telegram group. Okay. So please tell me which book should we follow for first prof. First year, I would say Harper. If you have just joined first MBBS, try reading Harper and Lippincott. Yeah, Harper and Lippincott. Method of teaching is just on another level. Thank you, Saurabh. I'm happy that you like it. So thank you all of you. So I will be sharing the PDF of this presentation in the Telegram group. Download the PDF and revise it. And please try to attend the test that is being conducted. That will be conducted on Friday. Okay. And continue watching the session. The same time, 7 to 7:45. There will be sessions till Thursday. And Friday there will be a test. Okay. So see you all. Good night.